Children, welcome to online classes of biology. So we were studying about various cell components, isn't it? And we have seen the endomembrane system. Okay, that includes what Golgi apparatus, ER. Okay, then we also have seen lysosomes. Okay, etc. Okay, and we studied about the mitochondria and chloroplast or plastids. Okay, what were the different types of plastids? There are three different types of plastids. What are they? Chloroplast, chromoplast and leucoplast. So chloroplast are plastids which contain colored pigments. Okay, they are having pigments. Which colored pigments? That is chlorophylls and carotenoids, isn't it? Chromoplasts, they contain different colored pigments, isn't it? Like carotene, xanthophylls, okay, etc. Carotenoid pigments are there in chromoplast, which gives color to the different colors to the plant parts, like petals, isn't it? Then some of the leaves, etc. And leucoplast are what? The plastids which do not contain any pigments, but they are helping in the storage of food. And I told there are different types of what chloro means the leucoplast. What are they? Amyloplast, leoplast, isn't it? Alluroplast. And we have seen also the structure of chloroplast, isn't it? Then now we are going to study about another organelle that is known as ribosomes. Okay, so ribosomes are what? Actually they are the site of protein synthesis. Okay, and they are seen as granules. Okay, granules like substances we can see in the cytoplasm. Understood? Then it is discovered or noticed for the first time by George Palade. Okay, and they are made up of RNA and proteins. Okay, and another important feature, they are not surrounded by any membrane. Okay, so these are the characteristic feature of what? Ribosomes. Clear? So ribosomes are what? They are the organelles which are involved in protein synthesis. And one speciality I told, they are made up of RNA. RNA is what? Nucleic acid, isn't it? So their function is what? They act as messenger, isn't it? And they means protein synthesis. They are acting as a connecting link between, you can say, DNA and proteins. But here you can see they are made up. This are taking part in the what in the ribosome structure also. Means ribosome is made up of proteins and RNA. So RNA functions, when we study, we are studying that RNA also can act as the structural component. Understood now? So here ribosomes. In ribosomes you can see RNA as the structural component. Clear? Then the eukaryotic ribosomes are 80S and prokaryotic ones are 70S. And each ribosome, they exist as two subunits. Okay, larger subunit and smaller subunit. Okay, and the two subunits of 80S means it is of eukaryotic cell, isn't it? It's of 60S and 40S, and that of 70S ribosome is the characteristic feature of what? Prokaryotic cell, isn't it? It is 50S and 30S. Larger subunit will be 50 and smaller subunit will be 30. Okay, so this S, okay, Swedberg's unit, it is the unit of it. What is it? Swedberg unit. Okay, Swedberg unit, and that is actually what it is for the sedimentation coefficient. Okay, sedimentation coefficient. It what it measures? It measures the density and the size. Okay, so Swedberg is the unit. Okay, standing for its sedimentation coefficient, and what indirectly it gives the idea of how much density and how much size is there. Okay, so both 70S and 80S are composed of two subunits. This we have seen. isn't it? Two subunits are there. And only during the protein synthesis they come and attach themselves to form a single unit, and there is no no membrane. So ribosome is an example for a for an organelle which is not covered by membrane. Is it clear? So ribosome's main function is to take part or to do protein synthesis. Next, we will study about the cytoskeleton. So, cytoskeleton means what? Network of filamentous proteinaceous structure. Okay, means in the cytoplasm you can see some what microtubules, microfilaments. Okay, intermediate filaments, filament-like structures. Okay, which are made up of what? Proteins, is known as what? Cytoskeleton. Okay, and this cytoskeleton, they do many functions like mechanical support, motility, maintenance of the shape of the cells. All these are the functions of what? Cytoskeleton. So what are cytoskeleton? They are filament like structures called what? Micro. It can be microtubules, it can be microfilaments. Okay, all this which are found in the cytoplasm. Okay, they are collectively known as what? Cytoskeleton. Okay, and they are doing different functions. Okay, like what? Giving support, helps in what? The movement. Okay, then it gives also shape to the cell. Okay, so then next what we are going to study is cilia and flagella. Okay, cilia and flagella. So cilia and flagella, how they differ? First, cilia are very small hair like outgrowths. Okay, they are small ones. Okay, and which work like an oar. Okay, means you would have seen this boards and all with oars, they'll be moved, isn't it? So in the same way they work as the oar. Okay, so in that way it help in the movement of cell. Then flagella, when comparing to cilia, they are longer and they are responsible for the movement. Okay, and bacteria when we studied, they are also having flagella, but they are different from that of the eukaryotic flagella. Okay, flagella of prokaryotes or bacteria is different in their structural organization from eukaryotic flagella. Okay, when you see the microscope, okay, through electron microscope, cilium or flagella, they are having a plasma membrane and the central core known as the axonium. Okay, axonium is the central core, and in the axonium you can see parallelly arranged microtubules. Okay, microtubules will be arranged parallel to the cilia or flagella. Understood? And the axonium, okay, the core consists of 9 doublets. 9 doublets means two. Okay, so likewise 9 groups you can see, 9 groups of two. They are arranged peripherally, they are arranged peripherally, and you can see in the center two microtubules, and this arrangement is known as 9 plus 2 array. Okay, so this central tubules, centrally also there are two microtubules. What are microtubules? They are proteinaceous rod like structures. Okay, they are made up of proteins and they are filament or rod like structures. Okay, and the central tubule, they are connected by bridge and they are also enclosed by the sheath. Okay, and each one of the what peripheral doublets, there are 9 peripheral doublets I told, isn't it? They are connected to the central ones by a radial spoke. Okay, so like this there are 9 radial spokes. Okay, and the peripheral ones, they are also connected by linkers. Understood? And this cilia and flagella, they are seen to be emerging from a another organelle that is known as centriole. Okay, centriole like structure, and it is also known as basal body. So here in this diagram it will be more clear. So you can see here the flagella or cilia. If you take the cross section, the outermost layer is known as the plasma membrane. Okay, this green type. Okay, plasma membrane, and inside that you can see the axonium. Okay, and the axonium, I told, there are what, microtubules. Microtubules arranged parallel to each other. Understood? So like this you can see a number of microtubules are arranged, and this each one of this. Okay, how many are there? Nine groups are there. 1 2 3 4 5 6 7 8 9. Okay, and in each group there are two microtubules. Okay, this is the cross section. Okay, so two are there, two microtubules are there. Okay, peripheral microtubules, and there will be two central ones, central microtubules. Okay, and they are connected by, they are also connected. Okay, the central two, they are connected. Okay, and then they are connected, they are covered by a sheath also. You can see a sheath that covers the central microtubule, and each of the microtubule from the peripheral doublet. Okay, among this, one will be connected to the center. Okay, one will be connected to the center through the linkers. Okay, or radial spokes. Understood? Radial spokes are there that connects each peripheral doublet to the central microtubule. Okay, and then I told the peripheral ones, they are also connected between each other. Okay, like this is the structure of what, cilia or flagella. So how you explain the structure of a cilia or flagella? The outermost covering is known as, the outermost part that covers the the flagella or cilia is plasma membrane. Okay, the core is known as what, axonium, and this consists of what, number of microtubules running parallel to each other. How many are there? 9, isn't it? 9 what, doublets means 2. You can see here, isn't it? In each of this, 2 microtubules are there. Likewise 9 groups are there, and they are what, radially arranged, peripherally they are arranged. Okay, and then also in the center there are a pair of what, microtubules, and that arrangement is known as what, 9 plus 2 array. Okay, the central tubules, they are also connected by bridges and is also enclosed by a central sheath. Okay, and the other tubules, the peripheral ones, are they are they are connected to one of the tubules of, okay, the central one through radial spokes. Okay, so like this, from the center each 9 doublets are connected, so there will be 9 radial spokes. Okay, and the peripheral doublets, they are connected by linkers. Understood? And the cilia or flagella, they start originating from the basal bodies. So about flagella and what, the cilia, you understood how it is available, and I told the microtubules helps in the movement. Understood now how by the movement of flagella and cilia they can move. Okay, then next what we have to study is centrosome and centrioles. Okay, so centrosome is actually containing two cylindrical structures, okay, which are arranged perpendicular to each other. Okay, so there are two centrioles in a centrosome. Okay, and this is the characteristic of what, animal cells. We have studied in animal cells centrosomes or centrioles are present, and it is absent in plant cells. Okay, and they are not membrane bounded, but they are bounded by a pericentriolar material. Okay, pericentriolar material is covering that, that is not the plasma membrane. Okay, and I told they are perpendicular to each other, they are arranged in the form of a cartwheel. Okay, and the structure, internal structure, if you see, they are also having what, 9 peripheral proteins. Okay, 9 peripheral, okay, evenly spaced peripheral fibrils of tubulin protein. This is one kind of protein only. Okay, tubulin. And each of the peripheral fibril is a triplet. There in flagella or cilia it was doublet, isn't it? Two what tubules will be there, but here you see it is a triplet, and the adjacent triplets are also linked. Here also 9 fibrils, microfibrils are arranged. Okay, 9 group, and in each group how many are there? 3 are there. Okay, 3 microfibrils made up of what? Tubulin protein. Understood? Then the adjacent triplets are also linked. The central part of the proximal region, that is in the center, there you can see a proteinaceous structure, and that is known as hub. Okay, and this part is connected to each of the tubules of the peripheral triplets by radial spokes, same like what the filament or the flagella or cilia. Okay, and the centrioles form the basal body of cilia or flagella. We have seen that. Okay, and that is one of the function of what? Centriole or what? Centrosome. What is it? They form the basal body of cilia or flagella, and it is also needed during the cell division. Okay, especially in animal cell division. Okay, spindle fibers are formed from this centrosomes. So that are the what functions of centrosomes or centrioles. What are they? They form the basal bodies of cilia or flagella, and another function is what? They help in the cell division. Okay, because spindle fibers, what are spindle fibers? Which get attached to the chromosomes, and it pulls the chromosomes to the sides. We will study this in detail in the cell division chapter. Okay, so understood now? So that time you can remember. Okay, so spindle fibers are there which is needed for the cell division, and it is originated from where? From the centrosome or centrioles. Okay, so today what we have studied is the ribosomes. Okay, and cytoskeleton, then structure of cilia and flagella, and we have also studied about centrosomes and centrioles. Okay, so we'll continue in the next class. Thank you very much.